Hello friends, welcome to this video. This is the second part of the question paper that we are discussing. We were discussing about the question paper of the Karnataka Examination Society or KPSC, which was conducted for the Assistant Professor Recruitment in the government first grade colleges. So we are discussing the botany question paper. We are talking the concepts related to it and giving you the key answers also. First 55 questions we discussed in the part 1. So if you haven't watched those questions, first, please, I'll give you the link in the description. Please do go watch that video, then come to the next 60 or 65 questions in this second part that I am going to talk about. Okay, we'll go to the video. So the first question over here, or the 56th question rather, is the correct palindromic sequence. So what is palindromic sequence? If you read it from right side to left side or left side to right side, it will be the same. For example, Madam, Malayalam. So all those things, if you read M-A-D-A-M, if you read it from left to right also, it's Madam. If you read it from right to left also, it's Madam, M-A-D-A-M. So such sequences are called as palindromic sequences. One important thing that you have to note down, they confused you by giving a gap in between is you have to read in case of DNA from 5' to 3' and then 3' to 5'. That is very important. See, if you look at this particular option, GAT and TGAT, these two are also palindromic, but that is not considered as a palindromic. The entire sequence should be considered like CGG, GCC. If you reverse it, you will get it as CGG, and if you read it from this side and if you read it from this side, both will be the same. Such sequences are called as palindromic sequences. Here you can see ATT it comes, but here AAT it comes, so it is wrong. Even this is not the option. This is just to confuse you. The given here. This is the right answer. This is the palindromic sequences. So in the next cases, in case of transcription of mRNA, what is the difference between an operator sequence and a leader sequence? They have asked. The difference is the operator sequences are not transcribed, but the leader sequences are transcribed. So that is the only difference between the operator and the leader sequences. Okay, here what they're asking you, it is the DNA replication itself. If you see the semi-conservative model of replication, you will find the leading strand and lagging strand. So based on that, the events they have asked. So the first thing what is going to happen is first thing is initiation of RNA primer. Your small strand of RNA primer is going to be synthesized by using primase. So this is the first step. Second thing, this RNA from this going to bind to this DNA from which the DNA is going to be replicated. So this binding of this RNA primer to the DNA is called as formation of RNA-DNA hybrid. That is your second one. Third one, what is going to happen is you're going to new DNA strands is going to be formed. That is the next thing. This is the third thing that is going to happen. It is like it is going to synthesize a new DNA strands from this DNA, from this RNA primer. That is your third step. Last step is whatever the Okazaki fragments are formed on the lagging strand, they are going to sealed by the DNA ligase enzyme. So based on this option is the right answer. So if you remember this DNA replication concept, it is very simple question, very easy to answer also. Moving on to the smallest unit of DNA which is capable of bringing a recombination is called as a record. Record brings a recombination. So next is the distance between the adjacent base of DNA. This is also very simple based on 12th standard syllabus it is. So if you see the double helical model of DNA, if you see the double helical model, the diameter or the distance between the two strands is 20 Armstrong. So that is 20 Armstrong has only two options. So you can cancel the other two options. This is all having 30 or 22 Armstrong. So this can be cancelled. And the entire one turn has 34 Armstrong and one turn has 10 bases. So 34 divided by 10. So the distance between each base pair will be 3.4 Armstrong. So the answer is option D, 3.4 Armstrong and 20 Armstrong respectively. Transgenic plants that can act as living bioreactors for the production of chemicals and pharmaceuticals. This is called as molecular farming. The medicinal plants are used in the biotechnological industries to extract the pharmaceutical drugs to pharmaceutical phytochemicals, secondary metabolites and all. That is by using biotechnological approaches like elicitation and all those things. So, or by upregulation of certain genes and also various biotechnological approaches or molecular approaches have been employed. So that is called as molecular farming. They're given two assertion and reason question. A feature of genetic code in which amino acid corresponds to more than one codon is called as redundancy or degeneracy. It is very true. So one for example, many amino acids are one amino acid might be coded by many codons. So that is called as redundancy or degeneracy. The acceptable pairing of several possible bases in an anticodon with the base in the third position of a codon. Yes, it lies in the third codon. That is what called as Obel hypothesis. So that is the right answer for option A also, for the assessment also. So both A and R are correct, and reason is the right. It is the correct reason for the assertion. So option B it is. Match the following with this list one thing. So here, see one thing, any one, if you know out of all this thing also, you can find out Haldane is the one who did all the evolutionary relation, phylogeny, evolutionary, all those things. So he was the one, JWS Haldane was the one who gave the relationship between the recombination frequencies and additive map distance. So based on that, it is option B, which is the right answer. And another assertion reason question is polyadenylation. Polyadenylation means addition of adenosine, that is called as polyadenylation. That is happens in the RNA modification when the DNA is getting converted to mRNA. So 3' to 5' strand will be there, and in the 3' end, what happens is the poly tail. So actually, if a strand, if you want to see. So if DNA is added from 5' to 3' end, in the 3' end, poly 8 tails are added. In the 5' you get a cap. You get a cap. Methanoid cap will be there. So in the 3' the polyadenyl tail are added, but here they have said it is 5'. So assertion is wrong. Reason is that a change in the expression of gene brought by which translocation to new set of genome results in position effect. That is true. However, that is not the right answer also. So A is false, but R is true. So that is also, it's not the correct reason, but A is false, R is true. That will be a right answer. Which one of the following is a phytoalexin? Phytoalexin are those chemical compounds that are produced by a plant after a pathogen attack. For example, a bacteria or a fungal or a virus has attacked a plant. In combating, in defense response to that, the plants will produce certain compounds called as phytoalexins. Pistatin is produced in your pea plant, and Danielon is produced in your papaya plant. Even Macayan is produced in certain plant. Bevistine is not produced. It's an artificial fungicide. It's a chemical fungicide. We use Bevistine even in our tissue culture laboratories too. It has an antifungal compounds. When I use Bevistine in my tissue culture media, MS media, fungal contamination will not be there. So in one of the videos I mentioned about that also, using of antibacterial compounds and antifungal compounds. So Bevistine is not a phytoalexin. It's a chemical. It's an fungicide. So that is also very easy. Cyclic AMP. So what happens? This is in the signaling pathways. If you have watched my GPCR video, in the GPCR video, I've said that cyclic AMP is getting formed by ATP. So it's a derivative. Cyclic adenosine monophosphate is a derivative of adenosine triphosphate. A simple question. Which of the following is correct? Pectic oligomers. It's an endogenous helicitous, and upon binding to specific receptor, it induces specific defense genes. It is true, and the defense gene will code for various enzymes and secondary metabolism, lectins and all, to fight against it. So both statement 1 and 2 both are correct. So when it's in the pectic oligomer, so the pectin compounds and all from the fungal bacterial cell wall, if it comes and binds to the receptors, the plant will activate its defense cascade. It triggers the defense responses in plants by activating all these genes. So both the statements are correct. A protein devoid of nucleic acid that can cause infectious disease called as prions. 12th standard concept or 10th standard concept. Very simple direct question. If a steel is split into distinct collateral vascular bundle, it is called as atactostele. So it is scattered vascular bundles in monocotyledons you can find. So I have made a video on stellar evolution in case of pteridophytes. If you had watched that video, this question also could have answered. So whatever almost I'm discussing here, 50 to 60% of it, all these things are covered in my videos, either practical videos or theoretical videos. So at least that knowledge, or at least once if you had watched all my videos in my channel, 60% of the answers of 60% to 70% of the papers, you could have easily solved it. So if you're watching the channel for the first time, please do subscribe my channel and watch the other videos, theoretical videos and all, conceptual videos and all. You have to watch it if you're appearing for any type of university examinations or competitive examinations and all. Okay, atactostele. During the entry of pathogen, a bulb-like structure is formed at the tip of the hyphae that is called as an appressorium. For example, as fungal spore is released. This fungal spore, when it falls on a leaf, this fungal spore will produce an hyphae. And in the tip, what will happen? The tip will be bulged. So this bulged portion is called as appressorium. From this bulged portion, a small protrusions will come like this, and that is called as your penetration peg. That helps in penetration. If they had asked which helps in penetration, then your answer would have been penetration peg. But here they have asked a bulb-like structure formed at the tip of the hyphae to increase the area of adhesion. So the answer is appressorium. So penetration peg is formed from the appressorium. It helps in the penetration and it penetrates deeper into the host cells. Triple response. This is in a hormonal. In ethylene, you would have studied in triple response. What happens? What is triple response means? You will get dwarfing of the shoots, thickening of the shoots will be there. So all those three things will be causing in triple response. Only this is not caused in triple response. So it's not a part of triple response. It's also very simple direct question. How many transmembrane alpha helices are present in G protein coupled receptors? So whenever if you watched, if I'm a regular viewer, you would have watched my GPCR receptor video. So many times I've said it is called as serpentine receptor, 7-pass receptor. So because it passes 7 times through the transmembrane, so the answer is 7. So 7 times it passes. That is why it is called a 7-pass receptor also. Which one of the following is an example for thick-walled dormant segregation of somatic hyphae that contains food reserves? Apothecium and all are the reproductive part. Sclerosium is the one that is having a thick-walled hyphae. All the hyphae in the fungal, what happens? It becomes thick-walled and it stores the food during some dormant stages. If the conditions are not favorable, it becomes dormant stage, and that is called as sclerosium. Okay. Preformed antimicrobial compounds present in the plants before the pathogen attack is called as phytoanticipants. Don't get confused with lectins and phytolexins and all. Lectins are the sugary compounds that are formed due for the when there's a rhizobium interaction, the formation of the root nodules. There the lectins are formed. Phytolexins, I said you, after a pathogen attack, phytolexins will form. Here you see they are preformed before the pathogen attacks the plants. As a strategy, they would have formed certain compounds. They are called as phytoanticipants. The plant may not become infected if its surface lacks pathogen receptors. So again, here I have ambiguity. Either the option may be A or D. Once the key answer comes, please cross check again. So if, for example, if there are no pathogen receptors itself in the plant, it cannot identify the plant, and the pathogen cannot come and bind to it to cause an infection. So based on that logic, I have given your option D, or it may be either A or D. Here also I'm having that ambiguity. Please check it. Please cross check it. Match the following with the correct combination. This is very simple. See, azola is used as biofertilizer. Everybody knows. Cyanobacteria, your algal bloom is caused by water bloom, or algal bloom is caused by secondary cyanobacteria. So that is also one of the answers you can write. And your alkaloids is your secondary metabolite, and corn is your mycoprotein. So if you know any one or two, this will become very easy. Based on that, the right answer is C. And moving on to the next one, which of the following is the denitrifying bacteria? The nitrogenous cycle you would have studied. Pseudomonas is the denitrifying bacteria, while all other three things, the nitrobacillus, nitrococcus, nitrosomonas, they're all nitrifying bacteria. So Pseudomonas is the denitrifying bacteria. In which of the following life cycle diploid stage is represented only by zygotes? So in algae would have been seen different life cycles. Haplontic life cycle means the haploid stage is dormant over there. Haploid is dominant over there. So your zygote diploid stage is represented for a very short duration, only zygote. So that is your haplontic life cycle. And the process in which the plant tissues are infiltrated with mineral rich fluids that precipitate, it is the petrifaction. All the minerals gets into it and all the living tissues gets decomposed, and all the living tissues, the gaps, the spaces are all petrified, or with the entry of it becomes mineralized tissue. That is called, it's a type of a fossil, that is called as petrifaction. Compression, mold, cast and all, you know what is a mold and cast and all. So that is all just a cast, that's our external feature. There will be no mineral entry, deposition will not be there. Petrifaction is the right answer. The branch of botany which explains the modern systematics on the basis of variations and evolution is called as, again here I have, because they're given two words. If they're given only evolution, then your answer would be biosystematics. If they're given only variation, then your answer would have been molecular systematics. I'm not very sure which is the right answer. So now, according to the rules of nomenclature, they have changed. Cruciferae has become Brassicaceae. Your cruciferae is nothing but it has crucifer. It has a plus-like structure. It has the flowers look like a plus. So that is why it is called as cruciferae or Brassicaceae. Cabbage, brassica, all those things, mustard, all those things comes in a cruciferae. Compositae is called as your Asteraceae member. Compositae, sunflower, daisies, triadax, all those compositae are called as Asteraceae, new name. because all the families' name should end with CEAE. That is the rule according to this IUCN. So they have changed all the older names, but these families are retained. Even the older names have become very famous. So we are maintaining even the older names. Umbelliferae is your coriander family. It is called as APAC, coriander and all. Palme is your areca nut, coconut, all those things. It is a Araceae member. So based on that, option D is the right answer. Binomy is accepted principal in nomenclature. However, Atibala, Nagabala, Mahabala not acceptable. Simple thing, even a 10th standard child will be able to answer this. They are not italicized, they're not Laticized, because we all know that scientific name should be either in Greek or it in Latin. The common names or the traditional names or any other Chinese names or Greek other names are not allowed. So they are not Latinized. That is why it is not accepted. The cell wall of fungi, very simple. Cellulose is a plant. Plant is made up of cellulose. Fungi is made up of chitin. Very, very simple. Fungi is made up of chitin. So main purpose of poisoning a plant specimen immediately after collection is what happens. We poison the plant specimen. It's like a disinfection process to kill the pathogens or to kill the insects. We poison before preserving as a specimen. So it is the option B, which is the right answer, to kill the germs present in the specimen. And this was a bit a tricky question because you have to know which family or which plant it belongs to. The herbals are given over here and they were given to taxonomists. You have to group them into how many families that was to belong to. So this was a bit challenging. If you should know the common names and the scientific names, then this would have been very easy. Chicory. So chicory is used in the coffee and all. Whenever making coffee powders, they had some chicory, right? Chicory is actually belonging into Asteraceae family. Colchicine is a chemical compound that is extracted from Gloriosa superba. You will bright red and yellow flowers. You will get it is found in the Western Ghats in Karnataka and all. It belongs to Liliaceae. And Amla, Indian gooseberry, belongs to Euphorbiaceae. Reserpine is obtained from Rauwolfia serpentina, that is your Apocynaceae member. Pyrethrum is obtained from your dairy flower, that is Asteraceae again. And so this both belongs to Asteraceae, single family, Liliaceae, Euphorbiaceae, and fourth one is Apocynaceae is Reserpine, and even Vincristine and Vinblastine is obtained from your Catharanthus roseus. Catharanthus roseus also has another name. I'm not able to remember now. Catharanthus roseus. From that it is obtained Catharanthus. So that it belongs to Apocynaceae. I mean, it is an anti-cancerous compound. So this is the one of the compounds. So fourth one is this both. So you have four families over here. Cotyledons, flesh and thicky due to the storage of proteins. Only Fabaceae member has this family of P, that is the Fabaceae. So that is the right option. Bicarpellary, syncarpous, unilocular ovary with single ovule. That is the family of sunflower. Sunflower is the Asteraceae. So that is the right answer. Bicarpellary, syncarpous, bilocular with axial placentation is the character of Solanaceae, that is the tobacco. And tricarpellary, trilocular, syncarpous with axial placentation is the characteristic of Liliaceae. Onion falls under Liliaceae. So according to this, the right option is option B. Another assertion and reasoning question. Agave and yucca have been placed in two different families in the earlier classification, but later it was placed in a single family called as Agaveaceae based on certain similar taxonomic attributes. And the karyotypes of the species of agave and yucca are identical. So both A and R are true, but this is not the right answer for that because there are certain taxonomical characters. They only said there are certain taxonomical characters are in common. This is not your taxonomical character. This is your cytological character. So taxonomy is based on the character, morphological characters, not based on cytological characters. So this is not the right answer for your assertion. So both, but A and R are true. So based on this you can find. Again, this one, the right option is not given over here. Which one of the following studies is concerned with the medicinal aspects of pollen grains, such as hay fever, seasonal asthma? Study of pollen grain is called as Palynology. Based on that, I eliminated criminology. Criminology will never come over here. Now, if the pollen grains are present in the honey, and if you are seeing the honey studies of pollen grains in the honey, that is called as Melito or Melisopalynology. This is with respect to honey, Melito or Melisopalynology. And geopalynology means the pollen grains that are found in the fossilized forms. In the petroleum products, in the fossilized forms, the term pollen grains will be trapped. That is called as geopalynology in the fossilized forms. So there is nothing called as Palynology. I don't know if there's spelling mistake or anything. So you will get a grace mark for this, I believe, because the 4 options are also not falling over here. In plumbago, what happens is the endosperm ploidy will be triploid because your haploid male gamete fuses with the diploid central cell to form a triploid primary endosperm nucleus. In the double fertilization, you would have studied triple fusion and syngamy. This comes under triple fusion. Syngamy is the fusion of an haploid female gamete with an haploid male gamete to form a diploid zygote. That is called as syngamy. The first fusion will occur over here. That is called as syngamy. Triple fusion. Triple means 3. So in triple fusion, what will happen? 3 will fuse. 2 central cell and 1 male cell will fuse to form a triploid primary endosperm nucleus. Therefore, the primary endosperm nucleus will divide to give rise to endosperm. So this plumbago is just to give the confuse the given here. So the triploid is the right answer. Consider the following statements. The correct statement: velamen is a specialized tissue found in the aerial roots of some orchids. It's very true. Velamen is a spongy tissue that is found for the absorption of the air. That velamen is a specialized tissue in the aerial roots. Lever mechanism is there in characteristic feature of salvia. This is also very true. There's a flower called as red color flower, salvia. An insect comes and sits over here. When it sits, it opens up. It's like a lever. So that is also true. Pappus is the modification of calyx. In Asteraceae, you can find pappus, small hairy root, hairy growths will be there. They are called as the pappus. That is also modification of calyx. It's very true. Gaspard Bauhund proposed a sexual system of classification. It was Carolus Linnaeus who proposed the sexual system of classification. So this is wrong. Acetobacter aceti is used for production of vinegar commercially. Yes, it is also true. Acetobacter is used. So based on this, option C is the right answer. It's very simple. A scientist recorded the following characters in a flower. Epicalyx is present in Malvaceae. Monothecous kidney-shaped anthers are also present in Malvaceae. Pentalobe stigma is also present in Malvaceae. And partially united corolla is also present in Malvaceae, hibiscus family. So if you see a hibiscus, if you remember one flower from each family, then this becomes very easy. So it is hibiscus family, Malvaceae, that is having this particular characteristic. So you can classify under Malvaceae. And this is also a very simple indicative statement which is not correct. Vallisneria in 12th standard you would have studied. Vallisneria is an aquatic plant, hydrophily occurs. Pollination is by water mechanism, that is hydrophily. Pollination in calotropes is by translator mechanism. You have pollinia. So pollination in calotropes is by translator mechanism. This is also correct. This is also correct. In potato, the pollination is performed in spiral bends to bring stigma in contact with the anthers. The self-pollination will occur. So this is also correct. But look at here, even if you didn't know this option C, option D, if you didn't know anything, at least one simple concept, if you understand, the versatile anthers. Versatile anthers means they are the anthers that are attached on the filament like this. So they will be moving up and down like this, that is present in the grasses and all. They are not found in entomophilous flowers. They are found in anemophilous flowers. They are found in anemophilous flowers. Why is that? Because the versatile anthers, when the wind flows, the anthers will move up and down. The pollen grains can be liberated with the help of wind. So anemophilous is with the help of wind pollination. So they are present in the anemophilous. That is the right option. They have asked you which is not the correct, so that is not correct. It is found in anemophilous. Very simple, this one also. Pellicle is found. Pellicle is a protein layer that is exerted in the dry stigma. This is a bit of a depth question. You should know what is wet stigma, dry stigma and all. And which one of the following is de-differentiation? So development of the callus from the parenchyma cell. Parenchyma cell is a permanent tissue. The permanent tissue again converts into meristematic tissue, and this meristematic tissue continuously divides. That is the de-differentiation meaning. So that is your right answer. Option C, meristematic parenchyma differentiating into callus is your de-differentiation. Which one of the following is the correct sequence of events during the course of pollination? This is also 12th standard level question itself. So you have to know that whenever pollination is the liberal or release of pollen grains, that is the first thing that will occur. The release of pollen grains is called as pollination. So you can see over here, nothing has pollination in the first stage. So you can eliminate all the option and choose the option B as the right answer. Pollination, then the released pollen grains either by insects or wind or water or snails or birds or humans or animals or bees, it will transfer onto the stigma and it will stick to the stigma. That is called as pollen adhesion. Then pollen hydration. What will happen? The pollen grains will take the water, nectar, sucrose, sugar, and all those things. In 12th standard, you would have done one experiment, the germination, pollen tube germination. So we supply certain compounds for the pollen grains will develop pollen tube. So that is called a pollen hydration. Then entry of pollen tube will happen into the style, and they will enter into the ovule, and through the synergies, it will burst open and it will fuse. That is the right event of fertilization in angiosperms. Then it leads to syngamy and triple fusion, both together called as double fertilization. That was identified or discovered by Strasburger in the plants. Very, very important. This may be another question. Who was the discoverer of double fertilization? Strasburger. Which one of the following is an alternate term for integumentary tapetum? Endothelium is also called as an integumentary tapetum. Direct question. Which one of the males is absent in the male gametophyte? The prothallel cell is absent. You have male gametes. The pollen tube develops into vegetative cell, generative cell, and this generative cell has these two gametes. So all these things are present. Prothallel cell is absent. So that is the answer for this question. The fertilization of egg and the secondary nucleus by gametes from different pollen grains is called as pollen tubes, is called as heterofertilization. Hetero means a different, not same. Heterofertilization, that is called as heterofertilization. Very simple. Ninth one, this is also another thing. If you had just applied little bit of logic, you could have easily answered this. See, they're asking you which of the enzyme are involved in the catalysis reactions that incorporate nitrogen derived from glutamine. So that nitrogen, it is involving in the nitrogen. So you have to have something related to nitrogen, transaminase, or it can be glutamine aminotransferase, because this also has amino, this also amino nitrogen. This does not involve nitrogen. So these two I can eliminate. And one more thing they have asked, nitrogen derived from glutamine. So from the glutamine, the nitrogen has been derived. So it is, of course, glutamine aminotransferase. So that is the simple logic. If it apply, you can easily answer this question. Which among the following is a photosynthetic bacteria having both PS1 and PS2? Simple logic. So again, this, if you had watched my videos related to photosynthesis, for PS1 and PS2, light reaction and dark reaction occurs. So if you had watched this video, that video could have easily answered. This PS1 and PS2 is present only in the green plants. If you see over here, the sulfur bacteria and the sulfur bacteria, they're all chemosynthetic, they're not photosynthetic. So both the PS1 are not present over here. And this is also on chemosynthetic bacteria, and it is purple, it's not green color. So the right option is blue-green algae or cyanobacteria. They are the one which can synthesize their own food, photosynthesize. So they have both PS1 and PS2. Gibberellic acid controls seed germination by breaking down of starch. So it induces the alpha amylase in which region of the seed they're asking. So they've given you the direct answer or clue in the stored starch. Where is starch stored? Starch is stored in the aleurone layer. So aleurone layer is the right answer. This is how you have to apply. See, so many concepts you can't study. So based on some basic concepts, you can answer certain questions. So those are some of the tricks that you can how to answer such multiple choice questions. You don't have to study the entire syllabus and go. If you're thorough with the basic concepts, you can study it. Indole 3 acetic acid is present in most of the plants. The structure is related to which of the amino acid? IAA is obtained, or the precursor for IAA is the tryptophan. So definitely its structure will be related to tryptophan. That is the right answer. If the lumen of the vessel in the wood is filled with the ingrowth of parenchyma tissue, it is called as stylosis. So if you have a xylem vessel, there will be ingrowth of balloon-like structure will be there. These are called as stylosis. These are generally found in the gymnosperm. Very important question this is. In which of the following plants successive increments, if you have studied anomalous secondary growth, so Borrelia will give rise to many vascular bundles. Successive secondary tissue is nothing but xylem and phloem. So successive, because of successive cambia, successive xylem and phloem will be there in the Borrelia. Anomalous secondary growth or abnormal secondary growth. That is your answer is Borrelia. Gums swell up in water due to imbibition. So this is also a simple physiological concept. Any substances which absorbs the water is called as imbibition. Gums are very dry. It absorbs the water and it swells up. So it is because of the imbibition. That is one of the important things. This is also another grace mark you will get, because all the four options are correct, but there's no option. There should be one option, all the four above. So all the options are correct. All these things leads to opening of the stomata. In the opening and closure of stomata, 12th concept, if you study potassium pump theory, there's something called as potassium pump theory. If you study the potassium pump theory, all the four options are correct. So this can be a grace mark again. Symbiotic nitrogen fixation. If you studied this, what happens? There are certain things called as nod genes in the rhizobia. that activates, and noduline genes that are present in the legume roots, those both needs to be activated, and there should be integration of the SIM plasmid into the root nuclear genome. So that helps in biological fixation or nitrogen fixation. And the sensing of plant flavonoids by rhizobia, the rhizobia does not sense the plant flavonoids, it senses the sugars and the lectins. If there are some sugary secretions, the roots will exudate. Root exudates will be rich in certain sugars and compounds that will attract the rhizobia in the rhizosphere. That is very important. So it's not the flavonoids. Flavonoids are actually defense. If root exudates start producing flavonoids, it will inhibit the microbial attack, or it will not allow the other plants to grow in that vicinity, surrounding vicinity. It's a defense mechanism. So it is A, C, and D. Based on that, option C is the right answer. CAM pathway, resolution acid metabolism, C3 pathway, C4 pathway, C5 pathway. So I have again made all these videos in my channel, I believe. So if you had watched that, this answer also could have easily answered. So CAM plants are able to fix the carbon dioxide in the dark. It's an adaptation for the xerophytic plants. So it's like opuntia, agave, pineapple, and all those things, cactus, all the plants, xerophytic plants are called as CAM plants. So they open the stomata during the night time and close the stomata during the daytime. So when they open stomata in the dark, they can fix in the dark. So this is option A is correct. They are able to fix the car in the dark, and they cannot survive longer periods in light. They survive for longer periods in light, because in the desert condition, you can see the light duration is very long durations. So, and also the CAM plants are not able to fix the carbon dioxide in the light, as I said you, because the stomata is closed. So option A and D, if you choose, that will be the option. That will be the right answer. Replacing thymine and guanine is an example of transition type of mutation. So if there's one particular nitrogen basis gets transferred, that is called as transition. When two or more non-allelic gene pairs affect the same character in the same way, it is called as polygenic inheritance. Poly means many, genic means genes. Many genes will contribute for a single character. Many genes, two or more genes, contribute for the same character. So from the question and the answer itself, there only it is your option. It is called as polygenic inheritance. It is called as following are the certain statements regarding the secondary metabolites in plants. All terpenes are derived from 6 carbon element. No, they derive from 10 carbon chain. So this is a wrong answer. Alkaloids are nitrogen containing compounds. True, all the alkaloids are nitrogen containing compounds. Pyrethroids is an insecticidal compound that is extracted from chrysanthemum. So it shows insecticidal activities. This also shows limno is a group of alkaloids. They have antiherbivoral activity. This option is wrong. So it's the option is only where B and C is present. This option is correct. Option D. Which of the plants following option match the plant's tissue type? See, it's very important, very simple. Tracheids are the chief conducting water elements in the angiosperm. Vessel elements are the chief conducting in the gymnosperm. Sieve tube elements in gymnosperms, and sieve cell in the food conducting of angiosperms. So sieve cell has all those things. So if you know that, you can easily answer. I think no, it is the other way. Tracheids are present in the gymnosperm. Tracheids are present in the gymnosperm. Vessel elements are present in the angiosperms. Vessel element includes your xylem vessel, xylem tracheid, xylem parenchyma, all those things it includes. Sieve tube elements are found in the angiosperm. Sieve tube elements, there are sieve cell, sieve plate, sieve cell, and companion cell, vast fibers, phloem fibers are all there. These are all called as sieve tube elements. So it is there in angiosperm, but the single cell is there in gymnosperm. So based on this, it is your option B, which is the right answer. Inheritance of ABO is called as multiple allelism or codominance. If the given codominance, that also is the right answer. So it's a direct question over here. In plant breeding, bagging is done for two different reasons. Bagging can be done. It is to also to avoid self-pollination, and it was also to prevent contamination. But usually what we do to avoid self-pollination, we cut the anthers. That is called as an emasculation. That is called as emasculation. So we cut the anthers, emasculation, we cut the anthers to prevent self-pollination. So more appropriate answer, the bagging is done to prevent contamination of the foreign pollen grains. To register a variety, the following criteria should be required. It should be a distinct variety, it should be a new variety, and it should be uniformly distributed. So if all these three are there, you can register a variety. A variety is a plant species or new varieties, we say, right? All those things. Double trisomy. So the name itself says double trisomy is +1 +1. So that is your double trisomy is represented, and even this answer I'm not sure. So if anybody finds out the answer for this, please post it in the comment for the others whoever are reading this question paper. It will be useful for them. 118, it is also very direct question. If you cross the F1 with a recessive parent, that is called as a test cross. If you cross an F1 generation with any of the parent, that is called as back cross. So either of the parent means it is a back cross. If it is a recessive parent, then it is called as your test cross. So it is very simple. Pure line breed refers to homozygosity condition only. It is called as pure line breeding. And new varieties of plants are produced by selection and hybridization. How do I produce a new variety? I have to hybridize and I have to select. It is very simple. Selection and hybridization. Which of the following are significant in maintaining the heat budget of the earth? It is nitrogen and oxygen, because they trap the heat and they help in the radiation and all. Because so that is why it is saying that nitrogen and oxygen helps in maintaining the heat budget of the earth. What is GPP of an ecosystem? See, GPP, rate of release of oxygen through respiration of a plant. When a plant is respiring, it will never release oxygen. So that is very important thing. It will never respire, it will never release oxygen, it will release carbon dioxide. Only in the photosynthesis process, oxygen is released. If you know the simple concept, you would have eliminated this question. I mean, this option. Next thing, net rate of carbon gain by the plant after respiration losses. So this is also somewhat wrong. Carbon is not gained, carbon is liberated. So this is also wrong. Total energy used to convert the sugar during the growth. So this also does not have something to do with GPP. So the gross primary productivity is the amount of oxygen that is released and when taking carbon dioxide. That is the photosynthetic fixation of the rate of atmospheric carbon dioxide is called as gross primary productivity, or GPP, of an ecosystem. So the concept of red data book is a book where they list all the animals according to the endangered and based on how vulnerable or extinct, all those things they mentioned. It is based on IUCN, International Union Code for Center for Nature and Natural Resources. That is very important. Initially you had IUCN, IUCZN, now it is IUCN. Code, International Common Nomenclature Code for Nature and Natural Resources, Conservation for Nature and Natural Resources, International Union for Conservation of Nature and Natural Resources. So based on this, the red data book is also governing. Then this is a simple cycle of nitrogen cycle. The events you have to understand. So if you know the nitrogen cycle, you can easily identify it. Nitrogen fixation will occur first, nitrification will occur, assimilation, then ammonification and denitrification will occur. This is the events of the nitrogen cycle. And the last one, the question of this paper, the energy flow in an ecosystem follows the law of thermodynamics. Exactly, it follows actually the second law of thermodynamics. The energy flow in an ecosystem is unidirectional, yes, and some amount of energy is lost when it is going from one trophic level to the other trophic level. Only 10% of energy is transferred, 10% law, you would have studied that. Rest of the amount of energy is lost in the form of heat or variable factors. So the both are option, both A and R is correct, but R is not the correct explanation. So both A and R is correct and R is not the correct explanation. So this is option C will be a right answer. So with this, we have finished 125 questions that was asked in the Karnataka government examination. So overall, I can say that if you know 70 to 80% of the basics, you would have scored very well marks in this. Or for your upcoming exam also, be thorough with exam, the basic level, 12th and degree level, you be very thorough. So only few questions will be asked, logical based questions and all. I'll make in the upcoming how to write competitive examinations like KSET, NET, or this is almost equivalent to KSET level. It is not even higher than KSET level. It's almost equivalent to KSET level, but it is lesser than NET level only. So you can easily answer such questions. So in the upcoming videos, I will discuss even more certain question papers. I'll give you certain tricks and all, how to solve questions and all. So please do subscribe to my channel and share this video with your friends and others. And please post your valuable feedback and suggestions in the comment sections. Thank you.